good morning, everyone who's in the Asia Pacific region. What time is it, Michael?
I don't know. It's just after 5 a.m. here, it feels like, but it's after 6 in the morning.
So it's a very good morning from Melbourne if you are joining us from this part of the world. But of course, very many people joining this PCF Prostate webinar are joining from completely different parts of the world. So good morning to all of you, and good evening and good day and whatever. My name is Declan Murphy, urologist at Peter MacCallum Cancer Centre here in Melbourne. It's my great privilege to co-host another one of these fantastic PCF prostate webinars, along with my legendary co-host, Professor Michael Hoffman, nuclear medicine physician and chief of all things prostate cancer at Peter MacCallum Cancer Centre. Michael, good morning.
Good morning, Declan. It's great to be here.
It is. So we're coming to you from the new GUcast studio. We run a podcast called GUcast. Do search us out if you haven't heard already. And we talk a lot about this topic this morning, PSMA imaging and theranostics, but we focus on all things GU oncology. So we're hijacking the GUcast studio to do our bit, the Melbourne bit of this PCF prostate webinar today, which I'm really looking forward to. What's the theme today, Michael?
The theme today is everything alpha. It's the hot topic and new targets, particularly a little bit of a focus on neuroendocrine prostate cancer and whether we can do something better than PSMA.
And I suppose the backdrop is it's been such an exciting time for radioligand therapy in prostate cancer the last few years with the betas, but we believe this is just the start of the journey in radioligand therapy in prostate cancer. And the focus of these webinars that we've been running with PCF for the past 3 years has really been to go a bit deeper into all of this stuff. We've had some really, really good webinars with key investigators and researchers from around the world over the past few years. They're all archived on the GUcast channel and the Prostate channel, so please go back and look. But this is a really delicious look to jumping beyond where we are with lutetium PSMA. And to that end, you've assembled a fantastic panel again, Michael, which we'll introduce as we go on. But we should, I suppose, introduce our studio team here as well today, shouldn't we?
We do. We've got a special guest, Dr. Marwa Rahimi, who's one of our radiochemists, one of the young investigators on our PCF Tactical Awards. So we really like to get the young investigators involved. And we've also got Dr. James Buteau, who's also a young investigator. He's doing a PhD and doing too many projects to talk about.
And if you're watching this on the webinar or backwards on YouTube, that's him sitting over there in the corner wearing his, what do you call that thing, James, on your head?
Well, it's a toque, the French-Canadian. I swapped the GUcast from last year for this Canadian version.
Toque. So it looks like a beanie to me, but James is there in the corner of the GUcast studio wearing his toque. All right, Michael, I suppose we should have a think about who else we need to introduce.
Maybe I'll just click to the slides to see how many people are online. So we had an amazing number of registrants. This is the global map with how many people have joined. At the moment, we have over 450 people online, and we had over 1,000 register from 74 countries, which is a prostic record.
Wow, that's very low. There's a
Here's
lot
a
of
map
interest.
of where people are from. It's quite extraordinary. And it's actually 75 because someone from Iran emailed me to say that wasn't on the list of countries in Zoom webinar.
Wow.
So they had to pick Turkey instead. So welcome if you're online.
Well, welcome to all our friends in Iran. Yep.
And then what sort of jobs do people do? The majority were identified as nuclear medicine doctors. But we've got a good range of people from industry, urology, radiation oncology, a lot of radiopharmaceutical scientists. We have some consumers online too. Just clicking. How do I go back?
Go back.
Go back. There we go. And then there was this other category. And there were quite a few others, so I was wondering what they were. And they were medical physicists. We had a lot of radiation safety officers. So that's interesting.
They're keeping an eye on you, Michael.
They're keeping an eye on us.
Yes.
And some radiochemists, interestingly, did not identify as radiopharmaceutical scientists. They thought they had a separate identity. These slides are auto-advancing. We might need to change that PowerPoint setting. And we have a lot of consumers online. So welcome, consumers, but we cannot give you medical advice today. For people online, you can post a Q&A at any time. Do that in the Q&A option at the bottom. Don't use the chat option. Use Q&A. And we'll also bring some people online onto the panel. So if you want to join the panel for everyone to talk live, either have your audio and or video ready, and during the Q&A time, we can advance you into the panel, and that'll be great. So and feel free to send me or James Buteau or Declan a message if you want us to move you in. All right.
Fantastic. So we do have a very, very good panel today, Michael, but we want to go over to our friends at PCF in California to welcome them, to say hello as well. So who have we got on the line first? It's Dr. Howard Sewell.
Thank you to the Peter Mac team for putting together this awesome group of investigators to talk about a topic that's very near to us. Last year, we were fortunate to have the resources to fund two $8 to $10 million programs in nuclear medicine that are both represented on this panel from Sloan Kettering with collaborators at Dana-Farber and et al. And of course, the Peter Mac group, all about alpha and all about new targets. So we're delighted to be here today. Declan and Michael, thank you for the opportunity and look forward to learning a lot.
Thanks very much, Howard, and always lovely to have you on the panel. And thanks, of course, to everyone at PCF for all their amazing support for what we do. And we have Dr. Andrea Miyahira as well on the line. Andrea, good evening to you.
Good afternoon in California.
Afternoon.
Good evening to whoever has evening going on in the world. I just want to echo Howard and say thank you to the Peter Mac team for putting on this amazing webinar, and also thank you to all of our panelists. I think it's a great program, and I look forward to everything I'm going to learn about alpha.
Yeah, for sure. Michael, so what's next?
So we're going to start with the Memorial Sloan Kettering team, and they're going to tell us about their tactical award. So we have a good mixture of radiochemist, medical oncology, and nuclear medicine specialist, all online. So that's great. We've got Michael Morris, who will lead off and share his screen for his talk. He leads the prostate and GU oncology program at Memorial Sloan Kettering. And also Lisa Baudet. Lisa, I know from my early days in nuclear medicine, because Dr. Baudet was doing everything related to neuroendocrine tumours with lutetium dotatate, which is how I got into the field. And I learned a lot from you, Lisa. So thank you for joining, and good to see that you've moved into prostate cancer as well. And we also have Jason Lewis, who is chair of the radiochemistry department. And he's joining us on leave. So thank you. So he'll chime in and answer the tricky questions that Mike and Lisa are struggling with. So welcome, everyone.
Fantastic.
Thank you.
We'll hand over to you, Mike, and the Memorial team. Thanks for joining us.
Thanks very much. And I'm hoping, Declan, that we get as freebies that very attractive GU Cast shirt that I see that you have, but none of your colleagues seem to have. So perhaps it's a special thing that we can share it.
Well, you know, like they all have some, but they just don't wear it. But I'm going to get you some GU Cast merch because I know you'll wear it, and when you're running around New York. Yep.
We'll put the merch on and spread the word. All right. Can everybody see my screen?
Yes, we got you.
Very good. So we're going to talk about new targets. And this work, of course, is really enabled by PCF and the generosity of the organization and its donors. And we'd like to just put that up front. Thank you very much, Howard and Andrea, for funding this research. These are Lisa's and my disclosures. And I'm going to start off with a figure that's from Misha Beltran's group, and I hear she's on the call. So Misha, I just want to credit you, and if there's anything that you think that we misstate, then you'll have an opportunity to correct. But what we're talking about is treatment emergent metastatic castration-resistant prostate cancer. And for those who may not be inculcated in the prostate cancer world, around 15 to 20% of patients who are treated with contemporary anti-androgen receptor therapy transform into, via a process called lineage plasticity, into a new form of prostate cancer, one which frequently isn't AR-driven, but could be. And this is characterized under a variety of nomenclatures, but for at least today's purposes, we're going to call it neuroendocrine prostate cancer. Neuroendocrine prostate cancer emerges via a whole host of a variety of mechanisms that look like they are triggered or accelerated by treatment with anti-AR directed therapy. And those mechanisms include loss of tumor suppressors, activators of tumor growth, and also epigenetic phenomena. And this process is very complex, very heterogeneous. But despite that heterogeneity, we're talking about a large group of patients at the end of the day who develop these biologic changes. And this is very poorly treatable disease. It's really difficult to actually find effective and durable treatment. And indeed, we would say that right now, there really is no standard of care for these patients, despite a lot of available therapies for others. So we're going to be talking about delta-like ligand 3. DLL3 is expressed in some neuroendocrine disease. It's actually quite copiously expressed in other more common neuroendocrine type disease like small cell lung cancer. In prostate cancer, though, it emerges really, at least the variety that we're talking about today in the treatment emergent context, well after the patient has been treated with AR-directed therapy. And you can see that in the top left panel, which Misha's group published a couple of years ago, in which you see the neuroendocrine expression come forth in the neuroendocrine group, but isn't really present in the adeno group, and certainly not in the benign or newly treated disease. This is true as well in terms of H-scores, and you can see it on our IHC grid here. If you look at the bottom right here at the neuroendocrine disease and under the DLL3 column, that is the treatment group that we're really talking about today, which is a singularly difficult disease phenomena to treat. And as you can see from these Kaplan-Meier curves representing the patients that these data were derived from, the folks with the DLL positive disease have a worse prognosis than the folks who have the DLL negative disease. Now, targeting DLL3 is something that's happening really across the board because this is a significant need in prostate cancer. There really are no treatments available, as I mentioned. And so immunotherapeutics are being developed against DLL3. This figure is from a bispecific antibody made by Amgen, AMG 757, but there are other companies such as Harpoon as well that have molecules targeting DLL3 on one end of the bispecific and CD3 on the other. So this is one approach. Another approach could be, of course, CAR-Ts. Another approach still that actually has been tested and already reported is Rova-T, an ADC. And some of the folks who presented this at ASCO 2021 with Rahul Agrawal as the presenting author, maybe on this call, it looked like it had modest response rates, some toxicities. But I would point out that all of the trials that are looking at these current approaches are unselected trials. And I think that by the end of this call, you'll see that there's an advantage to the theranostic approach in having an ability to select for the patients who are DLL3 positive, and then to deliver therapy that's targeted against those sites of disease. So with that, I'm going to now bring the microphone, so to speak, to Lisa, who's going to talk through some of the preliminary data that we have published on the theranostic pair that we are developing in order to treat DLL3 positive disease. And she's going to walk us through the next couple of slides. And with that, Lisa, why don't you take it away?
Thank you so much, Michael. And first of all, let me thank Michael and Declan for this kind invitation. I'm delighted to be here. So let me walk you through these few slides. So we've introduced DLL3 target, and So neuroendocrine prostate cancer lesions can be imaged with DLL3 targeted antibodies, which are the SC16 antibodies labeled with zirconium-89. So in this slide, you can see the results from our group where we studied cell lines exhibiting neuroendocrine differentiation like the H660. So they have synaptophysin positivity staining, whilst the LNCAP cell lines does not exhibit neuroendocrine differentiation and actually exhibits markers of differentiation such as the androgen receptor and the PSMA. So we have the availability of tracers to target these three receptors. We have gallium PSMA 11 for PSMA, we have zirconium DFO SC16 for DLL3, and dotatate for somatostatin receptor expression. So you see in this mouse model, you see the figure on the left part of the panel, as expected, no PSMA or somatostatin receptor expression, which is uptake, were observed in the H660 xenografts using imaging and immunohistochemistry. So you see the three panels below, and the lesion is on the left. Instead, these tumors expressed intense SC16 uptake, as you can see in the mid-low panel. Vice versa, the LNCAP tumors expressed PSMA and not DLL3, and dotatate actually was not very informative. So our group has tested the utility of zirconium DFO SC16 to selectively image DLL3 expressing neuroendocrine prostate cancer models, and they successfully demonstrated that DFO SC16 antibodies clearly delineates these tumors in subcutaneous xenografts. And as you can see from the histograms on the right, we have high targets over background ratio. So the tumor uptake is much higher than the uptake in the blood or any other organs. And most importantly, tumor uptake is significantly higher in DLL3 positive tumors compared to DLL3 negative tumors. So next, please. Next slide, please.
I'm hitting, there we go. Okay, go ahead.
All right, so this is preliminary results from our zirconium DFO C16 PET study. So these are two actual patients, and I believe that the images speak to the heterogeneity from themselves, to the heterogeneity in neuroendocrine prostate cancer differentiation and the heterogeneity of target expression. So the subject on the left has a metastatic NEPC predominantly diffused to the liver, with other lesions in the cervical thoracic nodes and the lungs. As you can see, the DLL3 uptake in the liver overlaps the uptake at the FDG PET scan. The patient on the right has diffuse metastatic disease to nodes and bones, and the uptake at the DFO SC16 PET, which is the DLL3 PET, as you can see, recapitulates the distribution of uptake at FDG PET. So, and I would also like to point out the low background uptake and the significant targeting of the tumor lesions allowed by the DLL3 PET scan. Now, the next one. And so the next logical step in our group, after demonstrating the presence of the target and the efficient localization of the targeting antibodies, was to develop a targeted radionuclide therapy following the theranostic principle. So we conducted a preliminary animal study in which we evaluated the lutetium version of SC16 antibodies, so lutetium 177 DTPA S616, in neuroendocrine prostate cancer xenograft bearing mice. The cells obviously are the H660. And we assessed the treatment efficacy and specificity at 3 different dosages of lutetium DTPA S16. So we have the 4.6, 9.25, and 27.8 megabecquerel per mouse. At all the 3 tested dosages, complete responses were observed, as you can see from the graph on the left part of the slides. So particularly if you look at the blue and the red curves, the 9.25 and the 27.8 megabecquerel curves were the curves that demonstrated that the dosages were curative. When we use the lower tested dosages, we obtain complete responses in some, in 63%, but we were successfully achieving a complete response with retreatment. So obviously this constitutes the basis for translation in human, also of the therapeutic approach. Thank you. Over to you.
Thanks so much, Lisa. So I'm just going to take our presentation home to its end now. First of all, I want to emphasize that what we are developing for prostate cancer will also have applicability for small cell lung cancer. And hence, on our tactical grant, we have John Poirier, we have Charlie Rudin, lung cancer experts, who will be able to translate what we develop from the prostate cancer world and this funding mechanism into the small cell lung cancer world as well. And therefore, we hope to benefit with the grant, not just one group of patients with prostate cancer, but an entire other disease, which represents its own significant healthcare issue. You can see that we have laboratory investigators, we have nuclear medicine scientists, and as well chemists. We have clinicians and Samir Zaidi, who's our junior investigator, who works in Charles Sawyer's labs. And we also have Felix Feng, who couldn't join us today, but providing a lot of the aim in terms of developing CRISPR models to optimize therapy. What we hope to do in the course of the grant is develop next-gen therapeutic and diagnostic radio tracer companions for full theranostic approach to the DLL3 positive prostate cancer patient, as well as the small cell lung cancer patient, to develop mouse models in order to better biologically characterize this disease state, and also CRISPR screens to understand what the controlling genes are in terms of DLL3 expression and how to best optimize those, as well as other therapeutics that might promote DLL3 expression and therefore enrich the target. Just to give you a preview of this is from Samir, who has been studying the biology from these patients. These patients are all biopsied after they are imaged. And you can see that even within this single cell data, that even within a specific lesion, that there are neuroendocrine positive, there are DLL3 positive and DLL3 negative foci within a specific lesion. So there's both interpatient variability and heterogeneity, intra-patient heterogeneity, and intralesional heterogeneity as well. And also he's showing the expression patterns that are of other markers that are associated with DLL3 positive and DLL3 negative foci of disease. So to conclude, what are the takeaway lessons from this? So first of all, neuroendocrine prostate cancer is a substantial healthcare concern because we're talking about 15 to 20% of all metastatic CRPC. It's a lot of men, and it's a lot of men without a good treatment option. But second of all, prostate cancer is actually not small cell lung cancer in the sense that we're seeing a lot more heterogeneous DLL3 expression. And I think that the images that Lisa just walked you through show how you can really assess within a given patient using the imaging whether crucial disease is expressing DLL3, that is hepatic disease, whether a patient that's progressing, whether those progressive lesions are DLL3 positive or not. But it's really important to understand that interpatient and intrapatient heterogeneity. The imaging will be key to a theranostic approach, and I would argue that our imaging data would suggest that those agents that currently are being developed in the immunotherapy world and the ADC world would do a lot better if there were an imaging companion in order to identify those patients likely to respond. Because as you can see, there's a lot of heterogeneity here, and you don't want a really good drug to yield a disappointing result simply because the correct patient selection criteria weren't applied. So I think that there's an overall drug development lesson for the neuroendocrine population. Finally, I'd like to advocate that we not try to characterize biologically what is a very, very broad group of patients that we're all sort of terming it neuroendocrine prostate cancer, but maybe start referring to these patients simply by the manifestation of the target, i.e. DLL3 positive prostate cancer, not try and subcategorize these patients into various groupings that we don't fully understand yet. And with that, I just want to thank again Prostate Cancer Foundation, and as well Declan and Michael and the whole group at the Peter Mac for allowing us the opportunity to present our data.
Thanks so much.
Amazing. That was stunning work. I have to say, I'm completely blown away.
Thanks, Team Memorial. It's a
Yeah.
really nice example of team science, because you just have so many disciplines working seamlessly together, and I think that's what PCF do extremely well, is bring all these different craft groups together to do amazing stuff. So we're going to switch tracks from DLL3 to choice of alpha emitters, and our tactical submission was together with UCLA, UCSF, and our colleagues in Essen in Germany, and our focus was on using lead-212 as an alpha emitter labeled to both PSMA but also novel targets, including DLL3. So we're not going to have time to go through our tactical in as much detail as the Memorial Group just did. So we're going to focus and narrow it down to just lead-212 and hand over to Marwa to tell us why we chose this unusual compound. It's a little bit unusual because, you know, in nuclear medicine, we shield radiation with lead. So to treat a patient with lead sounds a bit paradoxical.
I'm intrigued.
All
Marwa,
right.
yeah, good morning and welcome. Thanks for getting up so early in the morning and joining us.
No worries at all. I'm excited to be here.
Fantastic.
Okay, let's have a conversation about lead-212 and alpha therapy in general. So targeted alpha therapy, it's an innovative, very successful approach for the management of many different intractable cancers, and it relies on the overexpression of oncogenic receptors in cancer cells compared to healthy cells. So if we look a little bit closer at this concept, we see that the radioactive dose is delivered to the cancer cell using a targeting agent. These targeting agents are selected for their incredible selectivity and affinity to the, to the receptor target. So this is a very simplified version of the events that actually occur, but the key things I want to impress is the choice of the alpha emitter and the targeting agents. So I could talk at length about this, and I'm sure we'll have a discussion about this afterwards as well, but the focus of this talk will be just the use of lead-212 in targeted alpha therapy, its physical characteristics that make it really special as a candidate, and going through some of the challenges as well. But before we begin, I want to look at why are we using alpha in the first place? What gap is it fulfilling that beta emitters aren't currently? So the radiopharmaceutical properties of both alpha and beta emitters, they're characterized by two main features: their range and their linear energy transfer. It's basically a measurement of how powerful it is. Beta particles like lutetium-177, very well known to most of our audience, hopefully, they have a path length of about 10 millimeters, 1 centimeter, but they produce sparse ionization energy. Alpha emitters like lead-212, on the other hand, have a much narrower path length, about 1/10 of the beta, but they're 1,000 times more powerful. So these two, when combined, make the perfect complementary pair for therapy for patients. So you can treat patients initially, their medium to large tumors, by irradiating a large site with safe levels of beta emitters, and then chase that up with alpha emitters like lead-212 to target those individual cells, those microcancers that can arise. The alphas are able to deliver cytotoxic doses, lethal cytotoxic doses of radiation to the cells, inducing cell death. And at this stage, there's no resistance mechanism for this yet. So this makes alpha an incredibly promising approach for cancer therapy. Let's look at lead-212 a little bit closer. And for this, I'm going to benchmark it against actinium-225. Both of these target very similar cancers. Actinium-225 is the current golden child at the moment. So they can both exist in the same space. It's not adversarial. It's technically not a versus, but I think it's a good comparison to have in this. So lead-212 is generated using parents thorium-228 or radium, and there are generators available using both of these parents. Thorium-228 has a half-life of about 2 years, and so we can get generators that have a year shelf life, which is really great. Actinium-225, on the other hand, has two modes, but primarily through radiochemical extraction. The majority of it that exists in the world today is through radiochemical extraction. Unfortunately, because its parents are uranium-233 and thorium-229, there is overall a limited supply of it, and globally only about 68 gigabecquerels is being produced annually, enough for about 100 patients. Because of this limited supply, there has been investigations into accelerator-based routes using radium-226. This is a more cost-effective and scalable production route compared to radiochemical extraction, but there is, of course, the issues of using radium-226, and it also doesn't produce as pure an isotope. So let's look at compare their decay profiles a little bit as well. Both lead-212 and actinium-225 are what are considered in vivo alpha generators, in that once injected, they continue to produce alpha daughters as part of their decay. So lead produces a single therapeutically applicable daughter, bismuth-212, highlighted in orange, whereas actinium-225 produces 4 daughters. Theoretically, what this means is actinium-225 is a little bit more powerful. It produces 4 times as many daughters, so it should be able to deliver 4 times the amount of radioactivity. But on the other side, so it is able to do that, but it can also have a bit of a recoil, like a ricochet effect. With all of these alphas ricocheting off each other, it can sometimes irradiate neighboring tissues, maybe have some unfavorable accumulation in the kidneys, which is what we've seen previously. So let's look at another really positive aspect of lead-212. It's its clinically suitable half-life. Its half-life is 11 hours, which makes it really great for use in nuclear medicine. It allows for possibility of offering the patients outpatient treatment, as well as having really manageable waste. It's still alpha emitters, but it's a little bit more manageable due to its lower half-life. It's also able to deliver greater than 99% of its cell-killing radiation in only 72 hours. Finally, we can't end the talk without talking about some of the challenges. These, unfortunately, are not just to lead-212. This is a pretty universal challenges met by most alpha emitters. The first is the availability of these emitters for radiopharmaceutical research and the clinic. At the moment, demand is outstripping supply, but there are companies like Advanced Cell, Perspective Therapeutics, ArtBio, Oranomed, who are meeting these challenges. They're producing lead generators, or they're optimizing current lead protocols. There's also the potential for radiolytic breakdown of these generators. These alpha emitters produce magnitudes of energy, and so they can break down a few of the matrices inside the generators. And as always with any radionuclides, there's safety and handling protocols. We're hoping to get a lead generator established at Peter MacCallum Cancer Centre in Victoria, and so there are a lot of licensing issues that we need to go through in order to see that. But despite all of that, lead-212 ticks a lot of boxes. It has a lot of the physical properties that we really like, and in my opinion, it is first in class if we speak about alpha emitters. So that's it, everybody.
Thank you so much, Marwa. That was just amazing.
Apologies, we're having a couple little audio glitches.
Oh, did we?
It's
Sorry.
Declan's new GUcast studio. It's and some slide issues. I think if you can't see the slide big, you can click on your window that has the slide and picks. I think it's
Yeah, it was supposed to be spotlighted out, but Zoom Spotlight doesn't seem to be working this morning. So you can pin the individual window if you're not seeing it. But yeah, stunning stuff. LED's very exciting, I must say.
very exciting. So we are actively working on a LED 212 program, thanks to that PCF tactical grant. And we're going to have two more speakers. One is going to focus on translation of alpha to the clinic with an actual alpha PSMA study that's almost ready to go. And then we're going to talk to someone who's actually done some alpha therapy and get some experience. What is it actually like? So first up, we've got David Patterson, who's a nuclear medicine physician and also an endocrinologist, trained in both at Royal Brisbane and Women's Hospital here in Australia. And he is helping to lead one of the very first trials of LED 212 PSMA that I think is going to kick off shortly in his centre. So hand over to you, David. Please share your slides.
Great. Thanks, Michael and Declan. Yeah, thanks very much for the opportunity to speak to you this morning here in Brisbane. So I'll be talking about LED 212 radioligand therapy for prostate cancer. I'll just move that slide forward. And this has been covered in a previous talk, but essentially, you know, the real advantages of LED as opposed to other radionuclide therapy isotopes is, you know, as an alpha, it's got a high linear energy threshold. It's got a half-life that's, you know, well matched to the ligand half-life as opposed to, lutetium-177, or certainly in comparison to actinium-225, which has got a half-life of 10 days. There's relatively straightforward production capabilities, and there is, you know, increasing and growing supply. We're not there yet, but there's certainly, you know, a bright future with, significant quantities of thorium available for production, as opposed to, as we know, certainly with lutetium-177, there are, you know, issues. It's established production, but there are, you know, issues with global supply and demand at the moment. And there also is feasible SPECT imaging for dosimetry, which is not capable, which actinium-225 can't deliver, which is also appealing. When it comes to the LED 212 generators and the decay schema, this is just two of the different available products. So with AdvanceCell, that uses a thorium parent for production of quite pure LED 212, and Perspective Therapeutics is one of the other options which has opted for a radium-224 parent within that LED generator. As previously mentioned, LED has favourable characteristics really as an in vivo generator of bismuth-212, which is an alpha emitter, and in contrast to actinium, this just has it can decay either via polonium, delivering an alpha, or via thallium with another alpha emission. And implications for this mean that with a therapeutic activity sort of in the order of, you know, presumably 100 to 200 megabecquerels, it also provides the opportunity for imaging, which is not capable with a much lower dose administered or much lower activity administered with other alpha emitters like actinium-225. Just moving on, this is some of the preclinical data and which has been developed by Thomas Kreitzer, and I'm grateful for AdvanceCell for sharing this slide. But LED 212 ADVC001, which is the compound which is being developed and will be used in our trial, is quite similar to lutetium-177 PSMA-I&T, but with some minor modifications. And in this preclinical mouse study, it's shown that there is a significantly greater, tumor to kidney ratio in comparison to lutetium PSMA-I&T, which has much greater renal excretion. So this is hopefully useful to provide a greater therapeutic index. And we'll also credit Melissa Larter, who's and the Qtrace radiochemistry team at our statewide lab that have been working with AdvanceCell to bring this ready for clinical use imminently. And with regard to efficacy, again, this is preclinical data, again provided by Thomas Kreutzer from AdvanceCell. But this was a trial in mice. And essentially what it was comparing was there were 2 doses of either lutetium PSMA INT, or one dose, that's the green line here, or 1 dose of lutetium PSMA INT and 1 of lead-212 ADVC001. And the third arm was of 2 doses of the lead ADVC001. And you can see here, in terms of tumor volume, it was greater impact with the 2 doses of the lead investigational compound. In terms of survival of the mice after the administration, again, there was a statistically significant increased survival with the ADVC001 compound, and also less change in body weight of those mice. So that's the preclinical data. When we think about lead, it's good to know that it has been used in humans and has some therapeutic effect. Now, this is a different tumor type, but this was just recently published, a phase 1 trial of lead dotamtate in metastatic neuroendocrine tumors. Phase 1 trial up to 2.5 megabecquerels per kilogram, and really out of 20 patients, there was quite some exceptional responders, an overall 80% objective response rate. So that's sort of a proof of concept that as a targeted radionuclide therapy, it is effective. So looking to the trial that will be commencing imminently at the Royal Brisbane, it's a phase 1/2 dose escalation toxicity study of this lead compound, so lead-212 ADVC001. And essentially the trial inclusion and exclusion criteria are quite similar to the therapy study. And so it's got we've borrowed a similar name, the TheraPB study, but essentially it's in patients with metastatic castrate-resistant prostate cancer, prior androgen receptor targeted therapy and taxane exposure or contraindication. It uses similar PET criteria to the therapy study. So with gallium PSMA SUVmax greater than 20 at a site of disease and greater than 10 at all measurable sites, and also without FDG over discordant disease. Looking at the trial schema, there'll be a maximum of 18 patients with receiving up to 4 cycles of tolerated, and it's a relatively standard 3+3 dose escalation design. The initial 4 planned cohorts are at 60, 90, 120 and 150 megabecquerels. And there will be a period of safety follow-up, and there is actually some imaging components to evaluate preliminary response. Primary outcomes are dose-limiting toxicity and to determine the maximum tolerated dose. But as mentioned, there is some PSMA PET response and looking at clearance in blood and urine and significant adverse events. And just finally, to finish off my presentation, there's been some great work by our medical physics team in looking more closely at the potential for SPECT imaging of lead-212. And so this has just been some work showing that there is the capacity for imaging at therapeutic doses. And so this is just currently being built into the protocol, which is just delayed commencement of the trial slightly, but it should be ready to commence in the next month or so. And quite interesting, one of the things that Matthew's discovered is that you can actually image lead-212 at these administered activities using a PET camera, which is surprisingly via pair production from the very high energy gamma ray from thallium-208. So I might just leave it there and we can move on with the presentation. Thank you.
Thanks so much. Thanks so much, David. Look, alpha therapy with PSMA is not new. It's been done by our German colleagues for quite a number of years now. and also in many other countries, including by some groups in Australia. But I think it's fair to say that we still don't have any prospective data. All the experience to date has been compassionate access individual cases. So commend you on getting some prospective trials underway, because I think we really need that to move the field forward. And our next and last speaker before we have our Q&A is Dr. Megan Crumbaker from St Vincent's Hospital in Sydney. She works with Professor Louise Emmett, one of our nuclear medicine colleagues, and they've been doing a study of actinium-225, PSMA-617, and Megan's been kind enough to share some of the data on actinium to date, and some of her perspectives as a medical oncologist. So over to you, Megan.
Thanks for having me. So just to be clear, the phase 1 study that we're doing with the actinium is still underway, so I can't share any results, but there is previous data of this agent, and I'll present that, and then just some of my perspectives, just roughly from the patients I've treated. So, I'm not going to belabor this point. We've already talked about the perks of or the pros and cons of an alpha emitter. But specifically today, I'll talk about 225 actinium, PSMA 617. So keep in mind there are some antibody tracers going around as well, but I'm specifically talking about the peptide. So it does have a longer half-life than the lead that's been previously discussed. So it's got a half-life of 10 days, but with it being an alpha emitter, we're still able to treat patients as an outpatient despite that longer half-life. And we've talked about some of the advantages of an alpha emitter, the higher energy deposition and the shorter path length. And, potentially this is going to overcome radiation resistance in some of the lutetium PSMA resistant patients, may improve responses in patients with a lower PSMA expression, because you don't need as much of the agent to enter the cell to cause cell death. And perhaps it will be better at treating micrometastatic disease for these reasons. But I guess the other question is we know prostate cancer is very heterogeneous, despite the PET scan looking very homogeneous. When we do a PSMA PET, we know at the cellular level, there's a lot of heterogeneity. So with that shorter path length, are you going to miss out on kind of collateral cell kill of other cells that don't have the PSMA expression compared to a beta emitter like lutetium? So looking at the data that's already in existence for actinium PSMA 617, there is a meta-analysis of 6 retrospective studies, and we always know that retrospective data is not as high quality as prospective in general, but it included 201 patients across these 6 studies, and the PSA 50 response rate, so the patients that had a fall in their PSA by 50% or more, was 66%, which is similar to what we're seeing in a lot of the lutetium studies that we've seen in men with metastatic castrate-resistant prostate cancer. But the side effect profile is slightly greater than what we see generally in the lutetium studies, and particularly its hematological toxicity and the xerostomia. And again, particularly because of peptide, we get a lot of that uptake in the salivary glands. So the rates of xerostomia, more than 3/4 of the patients had xerostomia of any grade, and 3% had grade 3. Anemia in about 1/3 of patients, and thrombocytopenia in about 15%. And there were, not insignificant numbers with grade 3 toxicities with the anemia and the thrombocytopenia, which is, very substantial and potentially needing transfusional support. With the actinium, there's also been low rates of nephrotoxicity, particularly tubular interstitial process. And in this study, in this retrospective meta-analysis, there were 3% rate of grade 3 nephrotoxicity. There has been another study, again, retrospective data published on specifically men who have previously been treated with lutetium PSMA and then progressed. It's a very heterogeneous population. The definition of what lutetium failure or progression was not clearly defined. But in this study, I think this is really key because we're looking at the alphas as potentially something that can be used sequentially in men who have failed lutetium as another option if they still have persistent PSMA avid disease. In this study that presented data on 26 men, again, the PSA response rate, the 50% or more PSA fall was very similar, but these are small numbers. So obviously the range and the confidence intervals are going to be quite wide, but about 65%. PSA 50% response rate. So you're getting good levels of response. The PSA progression-free survival was only 3.5 months, but again, you're seeing a really big range here. And the overall survival was 7.7 months, which sounds low, but these were very heavily pretreated men, including, multiple lines of chemotherapy, androgen signaling inhibitors, such as abiraterone and enzalutamide, and also they all had lutetium as well. So very heavily pretreated. Keeping that in mind, these are heavily pretreated men. The rates of toxicity compared to the data in the meta-analysis are higher. So looking specifically at grade 3 to 4 toxicity. With anemia, we're talking about levels less than 80, and with the thrombocytopenia, less than 50,000. The rates of grade 3 to 4 anemia at 35%, that's similar to the rate of any grade anemia in that meta-analysis of the 201 patients. So that's really substantial. Most of those patients are needing blood transfusions and support. Leukopenia, common, but generally doesn't have too many sequelae for the patient. And then the thrombocytopenia, the grade 3-4 toxicity, nearly 1 in 5 patients. And then what's really key, and some of this might be due to the heavily pretreated nature of these patients as well, but I do think it truly is due to the treatment to a certain extent. And then what's really key is that all of the patients had xerostomia to a certain extent. And the xerostomia was actually significant enough to cause cessation of the treatment in nearly a quarter of the patients. And that's really, really important to consider. And I think, we oftentimes kind of downplay patients in their mind when you're consenting patients for these trials and you talk about dry mouth, they think, oh, that's not a big deal, compared to cancer and what it can cause, that's nothing. But I think one thing to consider is patients often at times do underreport the xerostomia, and I find in the clinic I really have to specifically question. So some of the data, especially this retrospective data, is probably underreporting on what the level of the xerostomia is. And then you need to think about the sequelae that really affect quality of life that occur as a result of the xerostomia. And there's actually quite a lot. So you get a lot of patients having anorexia, their mouth is so dry, they find it difficult to swallow, they're avoiding a lot of the foods they enjoy, or they get dyschezia and they have this awful taste in their mouth because their mouth is so dry. And this is, 24/7, and it can, it can be irreversible in most cases. The dry mouth can cause pain, discomfort, it can cause ulceration, it can cause dental issues. It's not a side effect to be downplayed. And I would say certainly my observation is much worse in my alpha treated patients compared to my lutetium treated patients. It's a very rare case where I have one of my lutetium patients complaining of a grade 2 xerostomia, whereas it's very common in these alpha treated patients. And keep in mind that when we're looking at the grades, we oftentimes report grade 3 as kind of, that's if it's low rates, that's okay. But grade 3 xerostomia means that they have this is the CTCAE criteria, inability to adequately element orally, they're being tube fed or on TPN. So, that's grade 3. So to say only 3% have grade 3, that's a big deal in patients. And you really have to weigh the pros and cons of that being a possibility. A lot are having grade 2, and that, they're needing to drink water all the time. They're waking up with a dry mouth. Their diet is, really modified. So, this is very, very significant, even a grade 2. So my perspective, I've treated a lot of patients with lutetium PSMA 617 or INT, and it's really well tolerated. And what I find is it generally maintains or improves quality of life when the patient responds to these treatments. We know there's the issue of durability of response in a lot of patients and treatment resistance, and alpha emitters may kind of overcome these issues in a subset of patients. But given the increased toxicity seen with the alpha emitter, and we are hitting the same target, we're hitting PSMA. I know there's a lot of talk of kind of tandem treatments and things with an alpha and a beta. We are hitting that same target. You're going to get overlap of toxicity, particularly in the salivary glands. I feel we really need a strong rationale to introduce actinium in patients. So patient selection is going to be key. I think its role really is going to be later on in the disease course, given the side effects seen. Potentially these patients with lower PSMA expression that are excluded from a lot of the lutetium studies, or potentially following lutetium resistance, where there's PSMA avid disease, because obviously a lot of these patients they have very limited options. So I think we're getting good responses, but we really need to get more data to figure out who we select for these treatments. And that's it.
Thanks so much. You can stop sharing your screen now so we can see everyone. Thanks for that. That was a fantastic overview. I might throw to a poll for the audience, so something will show up on your screen. You can answer. You don't have to answer it if you're a consumer and don't know. But the question here is, if we're planning a phase 1/2 study of a alpha PSMA, where should we position it? Should it be post-lutetium, post an androgen receptor pathway inhibitor and 1 or 2 taxanes like the VISION trial, post 1 taxane like the therapy trial, or should we just go post an ARPI and not require chemotherapy? We might give everyone a few minutes to vote on that, and then maybe value your comments, Mike Morris, on where you would position a phase 1/2 trial at the moment if you were opening one in your clinic. Yeah, maybe fire away. I think there's a bit of a mixture, which means there's no right answer. I think at the moment, leading is post-lutetium PSMA with 42%. And then second is therapy-like. But a lot of people going for it just post-ARPI, pre-chemotherapy, 22%. And only 13% going for a VISION-like population. Where would you position it, Mike?
I don't disagree that from a regulatory standpoint and drug development standpoint, its first place to be tested is going to be late in the disease course. But I'd venture to remind everyone that the place in which you could probably save the most life years and in which the physics of the alpha may work to greatest treatment benefit is in the micrometastatic setting, in the high-risk localized patient who's just about to undergo definitive local therapy or just following, so that you have a possible endpoint of cure as opposed to the disease burden and resistance that's already engendered in a patient who has widely metastatic disease.
Yeah, thanks for that. Megan, where would you position a phase 1/2 trial?
I agree with what Mike said. You always start these newer agents later on the disease course. When you're trying to figure out toxicity and efficacy, you really need to position it later on in the disease. I guess later on, once the phase 1/2 studies, the later phase studies, once you have more information on the toxicity and efficacy, you can rationalize putting it earlier on. But I guess as a medical oncologist, I'm always hesitant to say the word cure. It's very uncommon. So I think given the toxicity, I'd be kind of positioning it post chemo. I mean, chemo and prostate cancer is really well tolerated, and post an ARPI to begin with, until we get a lot more information on the longevity, like the duration of the xerostomia and some of these significant toxicities.
I thought I might get a Peter Mac perspective and bring our own Associate Professor Arun Azad into the panel for a few minutes. Where would you position a trial at Peter Mac, given our current portfolio of available treatments?
Yeah, thanks, Michael. I think, academically, probably almost it's most interesting to look in the post-lutetium space. I think there's a real unmet clinical need there. Megan summarized it very nicely in her last slide about, high responses and well-tolerated treatment with lutetium PSMA, but, durability of response and resistance is a challenge. So I think alphas have an interesting role there. I think that, depending on who develops alphas to phase 3, if it ends up being industry, sort of developed clinical development, then I suspect it'll be pitched late line. Mike alluded to this in terms of from a regulatory standpoint. A classic pathway in prostate cancer is to take drugs is to start late with drug development and then move the drug forward into more interesting spaces, like Mike has said. So, yeah, very interesting space.
And another question that I'm going to launch is, if we are doing an alpha trial, how should we combine it? Should we do it on its own, just alpha PSMA? Should we do it in combination with lutetium? There's a big vogue for either an alpha beta sequence or at the same time a PARP inhibitor. Immunotherapy or just give it on its own. Can people vote? And interested in your perspectives as well. Where should we position this? Is this going to be a standalone treatment, Mike?
No, I doubt it will be a standalone treatment at the day's end. And I think, your group, and as well the group at UCSF has already begun generating data on combinations with PARP and combinations with immunotherapy. So, and so the concept of RLT plus something is looking quite promising and tolerable in those contexts. Of course, your combinations are with lutetium, but I found especially provocative that data that came out of UCSF with just a priming dose of radioligand therapy with lutetium, followed by checkpoint inhibition, which really looked like there was some legs to the theory that you could have antigen release as a result of RLT and activate otherwise a relatively cold tumor. So I think we'll see what the data look like, but I think that all of these are really quite good, and it's hard to tell without data which will be the best, but I'm sure that all will be tested in combination with alphas as well.
Great. We might just go to the poll and see what people voted. There's really a bit of a split there. It's kind of everywhere. A bit of lutetium, PARP inhibitor, immunotherapy, single agent. It's almost like a quarter. So maybe we need to test them all.
Can I make a comment?
Yeah, absolutely.
I guess to me, prostate cancer, we know it's heterogeneous, from genomics and everything else we look at, including the data presented on the DLL3 expression. And we know in cancer, it's very clever, and you hit one pathway and there's crosstalk that opens up activation of another pathway. So to me, I just can't wrap my head around kind of hitting the same target two ways. To me, I think the way forward, and this is completely my opinion, is synergistic combinations rather than hitting that same pathway and potentially driving resistance without a counterbalance. But that's just my perspective.
So there's one.
Mike, can I? Oh, okay. Sorry, I was going to make a comment. Howard?
No, I was just going to say there's one idea that is nascent, but there will be soon a clinical trial. Lots of investigators are seeking ways to increase the expression of PSMA. And the group at the University of Washington, the Fred Hutch Cancer Institute, have discovered that HDAC inhibitors, and they've drilled all the way down, to the epigenome, to show how HDAC inhibition increases the expression of PSMA. And those clinical trials with lutetium-177 PSMA plus an HDAC inhibitor are on the eve of beginning. They're going to start. So that's another way, another whole brand new mechanism to think about when we think about combination trials with PSMA.
Thanks, Howard. And Jason, what did you want to add?
Yeah, I actually think the option you didn't add was all of the above, because I think that, you're classing it as prostate cancer, mixing every one of those additions has some really strong, pretty provocative preclinical data, whether it be DNA repair drugs or immunotherapy, that you can improve and you can enhance, not additively, but synergistically, the response, at least in preclinical models. But I think it's going to depend what type of prostate cancer you do that in. And I think that's what the question is. Lutetium actinium may be enough to get the bulk and then the metastatic disease. Making an immunologically cold version of prostate hot for immunotherapy could be the answer. So I think I'd have added an extra one in there. One, all of the above, depending on what type of prostate cancer you're dealing with.
And Dr. Baudet, do you have any opinions on this, given your really extensive experience, including in neuroendocrine tumors? I think there's a lot to be learnt from your sort of decades of experience with lutetium dotatate, and now what should we do with PSMA?
Yeah, well, so in principle, I totally agree with the panel. All of the above, depending on the clinical setting, that has to be discovered. There's one thing to be gathered from the experience with dotatate with the somatostatin receptor targeting theranostics, is that combinations have to be specifically devised not to increase toxicity, and to have a synergistic tumor effect, yes, but we know very well when we combined chemotherapy with somatostatin receptor targeting agents, sometimes the long-term effects were more prominent, particularly to the marrow. So that is obviously important in those protocols that are used in a much earlier stage of disease. Obviously, this is less important in much more advanced stages of disease. One comment that I wanted to make regarding the, let's say, induction of overexpression or re-expression, redifferentiation of prostate cancer, what is common to also neuroendocrine tumors is that HDAC inhibitors also promote, and we have a study in our department, HDAC inhibitors also promote the re-expression of somatostatin receptors, not just PSMA. So that opens up possibilities in all stages of the neuroendocrine, from the neuroendocrine differentiation to the earlier stages of disease.
It's an interesting perspective. Mike Morris, do you have any opinion on HDAC inhibitors? When I speak to my medical oncology colleagues, they tell me, oh, this always looks good in the cell work, but when you translate it to humans, you often see a very different picture.
Ironically, HDAC inhibitor testing began in prostate cancer. Kevin Kelly, who did the phase 1 study, he's a prostate cancer attending. He's now at Jefferson. So there is actually a history of clinical testing of HDAC inhibitors of prostate cancer who didn't move forward in prostate cancer, probably again because of the heterogeneity of mechanisms of growth in prostate cancer. But there's actually data and a trial, and it was the beginning of HDAC inhibitors in cancer.
Thanks. James, do you want to take us through some random questions? You can upvote questions, and then we can try and answer the questions that get the most votes.
Sounds good. So we have one that has been addressed, but to Marwa, how is lead-212 different or the same as other alpha emitters such as actinium-225?
No, I'm just going to quickly jump in there because if I didn't, my postdoc who tells this all the time would absolutely shoot me. But strictly speaking, lead-212 isn't an alpha emitter. Its daughters are. So every time we give the talk, if I don't stress that, I have a postdoc that says, no, it's not an alpha emitter, its daughters are. So just want to set that there as a radiochemist, the ground truth there.
No, you're absolutely correct. So it itself is a beta emitter, but it's what is known as an in vivo alpha generator. So it does have clinically relevant bismuth-212. Bismuth-212, I think, is the alpha emitting daughter.
Absolutely.
So in terms of their differences, I think it's look, there's risks associated with using any alpha emitter. I think that comes part and parcel with using it. But I think the fact that it does have just the single daughter, I think that makes it a little bit more stable to work with. I think its dosimetry is a little bit easier to deal with compared to looking at 4 alpha daughters in the actinium-225 decay profile. There's also issues with less of a recoil effect, less side effects there may be. So there's a number of different differences. That's not to say they're deal breakers. Actinium-225 has had a very rich history of being utilized in different clinical trials at the moment. It's, I guess, the golden child is what I refer to it. But I think long-term, if we were to build a future-proof platform, Lead-212, in my opinion, is the way to go, because there's no really issues with global supply. We've got many different companies leading the charge in developing the generators for use for research and commercial purposes. So some differences, not all deal breakers, but there's space for all of it.
There's a question here from Natasha Kendall. Has there been a comparison between Lead-212 and Actinium-225 or Terbium-161 in their ability to treat micrometastatic disease? Jason, are you aware of any direct comparisons?
Not all three. I mean, the comments were just made about lead, I completely agree with. There's pros and cons for everything, but in terms of that, the micrometastatic disease, I'm not sure I know. There's a lot of people now really delving into the radiation biology of these systems, comparing, say, betas with Augers with alphas. And whether or not, a mixture of those would be good, but I'm not aware, I could be wrong, of a lead versus, lead versus actinium versus terbium. I'm not sure, but, especially the terbium side, that's all pretty new, so I think that might still yet to be coming out publicly in literature. I would love somebody to do it and really look at these at the biological level, radiation biology level.
No, you're absolutely right. I think the few papers I've come across, they've modeled a lot of these. They've compared the data that's out there already, but again, you can't really replace experimental data, clinical data with theoretical, because theoretically, the alpha emitters should have the least side effects because they're so targeted. But from what we saw in the previous presentations, we're having really adverse side effects for the patients. So, we'll have to.
And I completely agree. And I think that also gets back to your comments about, chelates, conservation, momentum. When what are the ultimate fate of these daughters and how that makes a difference? And whatever parent nuclide you start from is the biggest determining factor. So I completely agree with all the things you said earlier.
Can I ask David Patterson a question? Because he's got his hands up, but I want to fire a question to him. One from the panel, which is no one's addressed the monoclonal antibody such as J591 with actinium-225, such as the Weill Cornell experience. Any thoughts on whether we should be using small molecule or antibody with an alpha?
Yeah, okay. Thanks, Michael. I might first just make the comment just relevant to that discussion about, actinium-225 versus lead. And I was just going to add, and I think just to elaborate for the audience about the recoil effect, that I think this is a really interesting and important element of Lead-212 for its potential toxicities, or hopefully lack thereof, is that with actinium-225, with that first alpha emission, the recoil effect can really shatter the bond with the PSMA, and then the whole sequence of subsequent alpha emissions following that over the next hour can occur. The products may diffuse out of the cell and lead to increased toxicity. Whereas with lead, that first emission is a beta particle, which has much less of an effect on that bond with the chelator, and so you then have significantly more bismuth 212, and then the subsequent alpha emission that's delivered should occur ideally within the cell and where the PSMA is located. So I think that's a really key and exciting element for hopefully preventing off-target toxicity. In terms of the comparison to the antibodies, I think one of the main concerns has been with with bone marrow toxicity, with the longer, residence time in the blood with antibodies compared to the small molecule inhibitors. So, at this point, yeah, I think my preference is with the small molecules like the current PSMA ligands, but I guess we'll see, time will tell.
All right, thank you. We have a lot of other questions, I guess about 17 minutes, so we can do a little bit of a rapid fire. I'd have one.
Mike was going to make a comment, I think, about the J591.
Yeah, I just wanted to say that we shouldn't get ahead of ourselves or ahead of the data in terms of deciding up front that the antibodies will have too much marrow tox. If you look at the data coming out of Scott and his colleagues group, they have dose escalated and found no heme tox that's dose limiting. And so they're now moving into their multi-dose studies and combinatorial studies. And so what we expect to happen, frequently we turn out to be wrong. And so I would give the antibodies a chance because actually their data is more mature and looks less toxic than the small molecule data that we have.
And also, Mike, to add on that, there are some targets like DLL3. All we have right now is antibodies. There are some targets which are not amenable at this point to small molecule or peptide-based systems. So, but antibodies are phenomenally good. Sorry, Megan, I didn't mean to jump in there. I didn't know if you were trying to jump in there, but antibodies work.
Yeah.
And I think the salivary toxicity is less, potentially.
Yes.
Much less, which is good for the alpha.
If I can add one brief comment to this discussion, antibody works, and the distribution of the antibody as seen at the diagnostic PET shows also the more or less blood pool uptake. And for example, you saw that DLL3 antibodies have very, very, very good target to background ratio. So that's also a guidance.
Right, I'd have a question for David again. So given the Therapy B trial that you're setting up, are there any radiation exposure concerns with the 2.6 MeV gamma coming from the thallium 208 from the lead-212 decay?
Yeah, look, not any major concerns. It's a reasonably low administered activity, and with that high energy, I mean, it's actually less likely to interact as well. So at this point, we don't have any major concerns for impact on radiation safety.
So I did note that there were lots of radiation safety officers that registered for this meeting, many more than last time. I think that's probably a marker of Pluvicto coming into the market and many centers establishing a theranostics program and suddenly encountering, oh, there's issues around radiation and administering this, particularly in high volumes. So is this going to be a problem with alphas? James, what do you think? Alphas versus beta and radiation protection precautions, bigger issue or smaller issue?
I think it all depends on which alpha you're using. So if you're using, for example, radium 223, doesn't matter. If you're going with, for example, with lead-212, there are more considerations for minimizing radiation to staff when you have like higher energy levels that you can get on a longer term basis.
But I think in general, there's an advantage to the alphas here because of their ultra short path length, and lutetium-177 or iodine has quite a bit of gamma emission. So both staff and relatives are exposed, but with alpha emitters, there's not a lot of exposure to other staff. So whilst they're more dangerous to the individual patient, they tend to be less problematic.
Just need a sheet of paper for shielding.
Just need a sheet of paper for shielding. That's right.
All right. There was a question about dosimetry also, I guess, for David. Do you have anything set up in the lead-212 around that?
Look, certainly we're hoping to gather some data on that. It's uncertain. We think that the the emissions for SPECT should be at the sort of limit for detection in tumour. It depends on how avid they are, but we are looking to do some dosimetry on sort of renal dosimetry and hoping to be able to image the salivary glands. But it's a question is with that dose escalation when that will become viable. But hopefully at a therapeutic dose, we anticipate we should be able to get some reasonable images for dosimetry.
All right, thank you. I have a question for Dr. Baudet. So can you speak a little more about the potential to treat neuroendocrine prostate cancer with radionuclide dotatate theranostics?
Obviously, this is the first thing that one thinks. You connect the word neuroendocrine and we think of somatostatin receptor expression, but that figure in the animal xenograph that we showed before, it's not just anecdotal, it's everyday practice. Neuroendocrine prostate cancer frequently expresses very heterogeneous amounts of or densities of somatostatin receptors, and usually the uptake at dotatate PET is low, much lower than the normal liver. So it's unfortunately not always a viable option. It's not, let's say, there is heterogeneity and there is variability, but as in general, typically, it's not the most viable of options.
Thanks. It's interesting. There's actually a study from Sherbrooke with Brigitte Garin, the 3-tempo triple PET imaging with PSMA, dotatate, and FTG. And essentially, of all those patients, none of them would have been potentially eligible either for dotatate treatment.
There's an interesting question here from an Australian, which says, when is lutetium coming on Medicare in Australia? And will they use the James Buteau criteria or a blanket bomb a la Vision USA style criteria? James, do you want to answer that?
Yeah, it's a great question. I think that I don't think we should close our eyes and treat blindly. It's just not the vision I have for theranostics. We do have some definitive data with therapy. We do know that the SUV mean on PSMA PET is predictive, so we can actually select and get good responses. We don't want to have underwhelming responses, and I think that MCRPC is very lethal. Those that we know have low uptake won't get good response from SUV means in that lower quartile, below 7. We can probably either get onto combination treatments or more active treatments such as Cabazi. So
I guess the question is whether, as we move to alphas, do we need more selection or less selection?
Can I just butt in, because the question about when are we going to get access in Australia, I had a meeting with Novartis Global yesterday, and they told me they submitted for TGA approval, regulatory approval in Australia last week, or 30th of June. So they finally started a pathway to get it into the market here.
Yeah, we really do look forward to more widespread global availability of lutetium PSMA, and there's a number of compounds coming through now. So it's happening. There's another question about what criteria we use for PSMA response. I might throw that one to Mike Morris. How should we be using PSMA PET to assess response?
That's such a hard question. And the caveat to that is response for what purpose? Is that clinical decision making? Is that a regulatory criteria? I think that there are clearly published criteria that set the table for looking at PSMA PET as a post-treatment endpoint in which to follow. What we have missing from that is the correlation of a given biomarker, whatever that might be, SUV mean, SUV max, distribution of disease, total disease burden. So what is the biomarker that is associated with clinical benefit, i.e. OS? So there's a big piece missing, the identification of the data element that is associated with the clinical outcome and what that association is. So I think that, Michael and others, Howard, have been very supportive of our efforts to work with regulatory agencies in order to have a roadmap to establishing what a response would look like on PSMA with regulatory recognition. But I'd have to say, we're not there yet. So we're still right now left with clinical trials that are collecting the data and the associations between PSMA PET and outcome. And we'll need to do better in terms of identifying a set or single element of the scan that we can call our leading biomarker to qualify.
Yeah, all our clinical trials are really still using CT and bone scan. And as nuclear medicine specialists, we report a lot of bone scans. And I think it's fair to say that we do find them challenging. They're not so easy to read compared to a PSMA PET. Lisa, do you want to comment just from a reporting perspective, trying to, formal criteria aside, trying to work out if a patient's progressed on a bone scan compared to a PSMA PET?
Yeah, I agree. It's not easy, but that's the direction that we are going to. There are several retrospective series which have been published by multi-centric groups from Germany, from Australia, from California. That show that there is a correlation, for example, with the interim PET and the survival parameters. There's now a very limited series, but very promising series regarding the end of treatment PET scan and the impact on survival. So I'm hoping that we can have prospective data which correlate better. Now, as to the relationship between the PSMA, the response at PSMA imaging and bone scan, obviously we still need data. As a nuclear medicine and coming from the experience of neuroendocrine tumors, it seems logical, since we are treating, if we're talking about lutetium PSMA, we're treating PSMA-avid disease. It seems logical also to investigate what happened to the PSMA-avid disease and restage with PSMA, but we certainly do need prospective data.
We've got Declan back on the panel.
Fantastic. What a fantastic webinar. I've thoroughly enjoyed this as a simple urologist, I must say. I really do feel that whole area of radioligand therapy is opening right up, and we're going to have to draw to a halt in a moment. I think, Michael, we're coming up to the end of the slot, and we all have to dial into the Peter Mac Tumor Board meeting in a couple of seconds as well. Finishing comments from you, Michael, and we'll throw back to Howard.
Yeah, it's been a fantastic webinar. It's a very hot topic, alphas. I think we're going to hear a lot more about it. Really excited to see some phase 1-2 trials coming through. No doubt we'll see some phase 3 trials following. I think, Howard, do you want to give us some closing messages or thoughts?
Well, my thoughts are that this is truly a dream team on the panel today. Thank you for putting this together, Michael. I too feel that the progress being made in radioligand therapy for patients is exemplary. Very early days, as Megan admitted, very, very early days, but trial by trial, we're learning more and more. And I would just urge the patients on this webinar, see clinical trials, because we're not going to move the ball down the field, whether it's rugby or American football, without participation in trials. It's really critical. So thanks to the Peter Mac and everybody else for their time today.
Thanks so much, Declan. There are many questions that we haven't been able to get to, and you're the social media guru. What's the best platform in 2023 to take those questions to? Because Twitter's going downhill a little bit.
It's become a bit miserable, hasn't it? But I haven't switched on to Threads or whatever. James, you're shaking your head. I don't know. I don't know. Maybe PCF should just set something up and we'll all fall into line as usual. But I think, there are quite a few good questions we need to get to. We will post this. There were some slides and audio issues. We'll tidy it up and post it as a podcast and a video podcast over the next few days. And if you follow Michael or Prostate or us, you'll find access to that. But then thanks again to all these amazing investigators. It's so exciting listening to this stuff. I've got to tell you, as a simple clinician clinician urologist looking after prostate cancer patients full time, I'm just incredibly excited by everything that I've heard this morning or this evening. So that's it, Michael.
Wonderful. We'll do it again in another few months.
We will, and thank you all very much for joining us, and we wish you all a very good day. Thank you.
Thank you.
Thank you.
Thank you. Bye.